So we've learned about the structure of DNA. The next thing we need to learn about is how DNA can make copies of itself. And this is something that your body does every day, your cells do every day, and there's a reason for this. So before a cell divides, it has to make a copy of its genetic material. So why would a cell need to divide? Well, our cells in our body, and any organism's body, they get old. So we have to make new cells. We have to, if we get injured, we have to make new cells to repair what's been damaged. So we constantly are having to make new cells for various purposes. But if you think about it, when we make new cells through our old cells. So our old cells will split and make new cells through a process called mitosis, which you should check out and learn about. Um, but this process basically splits the cell in half to make new cells. But you have to think about it with DNA, that would not be a good thing. A cell does not want to give away half of its DNA because that's half of its instruction manual. The cell doesn't want to give away half of its instruction manual. That would not be good. So in order for a cell to make a new cell and to give DNA to that new cell, the cell needs to make a complete copy of its DNA so it can pass a copy to the new cell and keep its own copy. So that's why we have to do this process called DNA replication, where DNA makes a complete copy of itself so that it can be passed on to a new cell. All right, so that's what this sentence means. Before a cell divides, it must make a complete copy of its genetic material to pass on. And this process is called DNA replication. Watson and Crick, which were the two guys that discovered the structure of DNA, it's such a huge finding back in the day. But this is a quote from them saying, each strand has all the information needed to reconstruct the other half by the process of base pairing. And you've already learned about base pairing or complementary bases. A always goes with T, C always goes with G. So if you know one side of the DNA, it's easy to make the other side because you just do the base pairs. So it's a really awesome, efficient process because each strand has all the information needed to make the other strand, which is awesome, and you'll see this come into play. So here's a very simplified view of DNA replication. Obviously here we're only showing 5 base pairs. You have to think that your actual DNA molecules have millions of base pairs. So this is very simplified. But let's look at this DNA molecule right here. You have 2 strands, of course, because DNA has 2 strands. And this is called the parent DNA because it's the original DNA. This is what we're going to start with. The DNA strands are then pulled apart. We have to pull them apart so we can build new sides onto the old strands. So the parent DNA is pulled apart, and then you can see one side comes up here, one side comes over here. They don't actually move. This is just showing kind of what happens and giving room to show what's going to happen. So here's the parent strand, and a new strand is going to be built on to the parent strand due to base pairing. So A goes with T, C goes with G, G goes with C, A and T and A. Same down here. Here's the other parent strand, and you're going to have new strand built on. And this is what you're going to end up with here. So you have 2 identical DNA molecules that are exactly the same as the parent DNA. You can look at the structure, excuse me, the sequence, they're exactly the same. You got AT, CG, GC, AT, TA. Same here. Same here. So you started with 1 DNA molecule, and now you have 2 DNA molecules that are identical to the first. The only difference is that the new DNA molecules have half old and half new DNA. They have one side that's the parent strand and one side that's the new strand that's actually going to be called the daughter strand. Okay, same here. You got parent strand, daughter strand. So that's actually called semi-conservative replication because the new DNA molecules are half old and half new. So that's just some terminology you need to understand. Semi-conservative means the new DNA molecules are half old, half new. So let's go in a little more detail to this process. The DNA is actually split apart due to this protein here. Again, proteins are the workers of the cell. And this protein is called helicase. And helicase's only job in the cell is to split apart the DNA. It kind of acts as a zipper and unzips the DNA. So you can see it's moving down this way and it's separating these 2 DNA strands. It's separating the double helix because we need to separate it to get to the strand so we can build new strands. The next step brings in another protein called DNA polymerase. And this guy's going to kind of be like a builder. He's going to build the new sides of DNA onto the parent strand. So remember, we're building new DNA called the daughter strand, and we're using the information from the parent strand to build that. And that's exactly what DNA polymerase does. He's kind of like Bob the Builder. He builds the other side of the DNA. So the first step, helicase, breaks apart the DNA, and now DNA polymerase is building on the new DNA. And then in the last step, DNA polymerase, even though it's very good at its job, sometimes it leaves these little gaps, as you can see here in the DNA. And even though DNA can do these really cool things, or DNA polymerase can do these cool things, it cannot seal these gaps. So you have to bring in this other protein called ligase that's actually going to seal together these gaps so the DNA is completely connected. You need the DNA to be completely connected. You don't want to have any breaks in there because it'll make it unstable. So ligase kind of just follows behind DNA polymerase and seals in any gaps. It's kind of like glue. And you can remember ligase is the glue because ligase has a G in it, and none of the other proteins that we talked about have a G in it. So this is your glue right here, is your ligase. So let's go through these 3 steps again, because you need to know all 3 of these steps, and you need to know the names of the proteins that do each of these steps. So the first step again involves the protein helicase, and helicase acts as a zipper to unzip the DNA, to split apart the DNA. Okay? DNA polymerase is kind of like Bob the Builder, and he's going to build on the new sides of DNA. And notice he's working on both strands of DNA. He's not just working on one. You work on both at the same time, so it's more efficient and you get the process done quicker. So DNA polymerase is the builder that's going to build the new side. And then lastly, ligase follows right behind DNA polymerase and seals in or glues any gaps that are left between the DNA. So those are the 3 players that you need to know, the 3 proteins that are involved in this process. Because DNA is not a worker. It can't do this process on its own. It can't replicate itself. DNA is just a bunch of information. It doesn't actually do work. Protein are the workers. So the proteins are the ones that actually make this process happen. So you need to know these proteins and how they're involved in the process. This is just a quick video that I want to show you. Again, this is very simplified. This process is actually way more complex than we're even talking about, but this is, you don't really need to understand the more complex details of this, but this is just a very simple view of DNA, excuse me, DNA replication, but I like that you kind of actually get to see it happening in an animation. And it talks about helicase and DNA polymerase, but it doesn't even talk about ligase. So this is what you need to understand from this.
DNA replication is defined as the process by which an organism's original DNA is used as a template for the production of a new complementary DNA strand. An enzyme called helicase unwinds the original DNA's double helix, creating a replication fork. Next, an enzyme named DNA polymerase III works down the leading strand and up the lagging strand of the replication fork, synthesizing 2 new strands of DNA by taking free nucleotides and pairing them with the complementary bases on the original DNA template. The process of DNA replication is described as semi-conservative because the 2 copies of DNA produced each contain 1 strand of the original DNA and 1 entirely new DNA strand.
Okay, so that just kind of shows you in motion how this process works. So let's go over what we talked about. You have your parental DNA, that is what you're starting with. They get split apart by helicase, and then you build on daughter strands or new strands using DNA polymerase, and then ligase seals up any gaps, and that leaves you with 2 identical DNA molecules that have half old, half parental strand, and half new, half daughter strand. So parental strand is from the original strand, and daughter strand are the new strands. So you need to know that terminology. Here's your complementary bases. Again, complementary just means that they match together. So A goes with T, C goes with G. It's just a fancy word for matching. Semi-conservative means that you, in the new DNA molecules, you have half old and half new. So half the DNA is from the parent strand, half is from the daughter strand. That's basically what this says right here. And then just definitions of parent and daughter. So parent strand is from the original, daughter is the new strand. And I just want to wrap up about why this process is so cool. So one thing is the sequence of bases, and we already talked about this, but the sequence of bases in the parent strand provide all the information to make the daughter strand. It serves as a template, and a template is just kind of something that you can follow, so like instructions. And the parental strand actually serves as instructions to make the daughter strand, which is really awesome and makes this process very efficient. Rather than just building something from scratch, all the information is there to build the new strand. Also, DNA polymerase, which is Bob the Builder protein, not only does he build the new strand, he actually also acts as a proofreader, which means as he's building, he kind of checks his work. And if he finds that he actually matched the wrong base, so let's say he came to an A and he put a G instead of a T, he would back up and fix that before he moved on. So he kind of checks itself before there's a mutation or something wrong in the DNA. And 99.9% of the time, he does that well. He proofreads and finds the mistakes. We will learn down the road about mutations where every once in a while he does miss a mistake, okay? But that's something you'll need to know down the road. But this is basically all you need to know thus far about DNA replication. And we will move on to the next topic coming up next.